Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this fatty acid oxidation lecture, I will be explaining you regulation of fatty acid oxidation. Let's get into the details. When it comes to regulation of fatty acid oxidation, regulation of fatty acid oxidation is revolving around a single molecule that you must remember to remember the regulation of fatty acid oxidation, and that is malonyl-CoA. Make sure you remember this name of this molecule, malonyl-CoA. How do you get malonyl-CoA? Malonyl-CoA, it is basically coming from an acetyl-CoA. Acetyl-CoA is a 2-carbon molecule, as I am showing here. Here it is, acetyl-CoA, which is a 2-carbon molecule, will be converted to malonyl-CoA, and this will be done by enzyme acetyl-CoA carboxylase. Now, acetyl-CoA carboxylase is the one that will be converting acetyl-CoA into malonyl-CoA, and this reaction will be going on whenever person is in fed condition. So under fed condition, fed state, as you know, there will be release of insulin, and this insulin, especially in the liver, it is going to induces this acetyl-CoA carboxylase, and also it is going to keep this enzyme under dephosphorylated state. Thereby this enzyme is active, and when this enzyme is active, it is going to convert more and more acetyl-CoA into malonyl-CoA, so that malonyl-CoA is going into fatty acid synthesis. In fact, the purpose of making malonyl-CoA is to make a fatty acid synthesis. A fatty acid is synthesized using a malonyl-CoA. So that's what is the purpose of insulin. While this malonyl-CoA is elevated, when the fatty acid is synthesized, fatty acid oxidation should not go on. Remember this. Because fatty acid synthesis and fatty acid oxidation, that is beta-oxidation, these are antagonistic pathways. Always antagonistic pathways should not run simultaneously. That's why whenever malonyl-CoA levels are high in the cytoplasm, this malonyl-CoA, it will have a negative effect on a CPT1 enzyme, CPT1. The CPT1 enzyme, it is there over outer mitochondrial membrane, and it is involved in the transport of long chain fatty acids into the mitochondrial matrix by carnitine esterification. So CPT1 over outer mitochondrial membrane, what it does, it is going to add carnitine to fatty acyl-CoA to make it as fatty acyl carnitine. We have seen that in our previous chapter, where mechanism of transport of long chain fatty acids into the matrix of mitochondria. So whenever fatty acid synthesis is going on, at the same time fatty acid oxidation should not go on. Remember that. So malonyl-CoA, when it is synthesized at high concentration in the cytoplasm, so the malonyl-CoA, what it does, it is going and inhibiting an enzyme which is located over the outer mitochondrial membrane. That name of the enzyme is CPT1. So the CPT1 enzyme, let me write it down. CPT1 enzyme is involved in the esterification of fatty acyl-CoA with the carnitine to make fatty acyl carnitine, and the long chain fatty acid, that's how it is transported inside the matrix of mitochondria. We have seen that in our fatty acid oxidation lecture in the beginning. Now, whenever malonyl-CoA is inhibiting this CPT1, so that means long chain fatty acids are not transported into the matrix of mitochondria. That means beta oxidation do not go on. So when fatty acid synthesis is going on under fed condition, beta oxidation should not go on in the matrix of mitochondria. That's how it is regulated. So the malonyl-CoA, it is kind of controlling the beta oxidation by not allowing long chain fatty acids to go into the matrix of mitochondria. Okay, now that happens under fed condition. So insulin will make sure malonyl-CoA levels are high, and that malonyl-CoA will have a negative effect on CPT1. And also under fasting condition, what will happen? So whenever person is in fasting condition, fasting state, so fasting hormone is glucagon, And this glucagon, what it does, it is going to have a negative effect on acetyl-CoA carboxylase enzyme. Basically, glucagon is going to phosphorylate acetyl-CoA carboxylase. And when acetyl-CoA carboxylase is phosphorylated, it is no longer converting acetyl-CoA into malonyl-CoA. When acetyl-CoA is not converted into malonyl-CoA under fasting condition, so the levels of malonyl-CoA will decrease. And when the levels of malonyl-CoA decreases, so its negative effect on CPT1 is not there. That means CPT1 is working because the negative effect is not there. So fatty acids, long chain fatty acids are going through the inner mitochondrial membrane and undergo oxidation. So at the same time, when the fatty acid oxidation is going on, fatty acid synthesis will decrease because malonyl-CoA levels are less. Malonyl-CoA is the one that has to go into fatty acid synthesis. So like this, glucagon will increase beta oxidation of fatty acid by keeping malonyl-CoA levels low, and insulin is going to keep fatty acid synthesis high, beta oxidation low, because malonyl-CoA levels are elevated. So like this, it is all around malonyl-CoA concentration that you must remember. This is in fed condition and fasting condition. Now let's move on to see what happens whenever a person is exercising. During exercise, like activity, especially in the skeletal muscle. So in the muscle, how this fatty acid oxidation is controlled. So in the skeletal muscle, AMP levels will increase whenever a person do exercise. So this AMP is going to activate AMP activated protein kinase, and thereby it will have a negative effect on acetyl-CoA carboxylase. Let's look at that in the next slide. So here is a regulation of fatty acid oxidation in the skeletal muscle and cardiac tissue. This figure is showing you how a fatty acid oxidation is regulated in the muscle here. Now the AMP, whenever muscle contraction is going on, so the AMP level rises, and this AMP is going to activate a protein called AMP activated protein kinase, and this AMP activated protein kinase has two roles. One of the role of AMP activated protein kinase is to keep low malonyl-CoA levels by not allowing conversion of acetyl-CoA into malonyl-CoA. You are seeing that in a number 1 reaction here, right? So the number 1 is ACC, ACC2 is acetyl-CoA carboxylase, is written as ACC2. Here is the acetyl-CoA carboxylase, ACC. Now, acetyl-CoA carboxylase is negatively modulated by AMPPK, that is AMP kinase, AMP activated protein kinase. It is going to phosphorylate acetyl-CoA carboxylase and keep the enzyme activity low. Thereby, acetyl-CoA is not converted into malonyl-CoA, and that's one thing. The second thing is same AMPPK, that is AMP activated protein kinase, it is going to act positively on another enzyme called malonyl-CoA decarboxylase, and that malonyl-CoA decarboxylase is written here as MCOADC, MCOADC. MCOADC is malonyl-CoA decarboxylase. So what this enzyme do? As the name says, decarboxylation. It is going to decarboxylate malonyl-CoA into acetyl-CoA. Thereby, it decreases the levels of malonyl-CoA. So like this, increased AMP during muscle contraction, activate AMP activated protein kinase, that's going to inhibit acetyl-CoA carboxylase, thereby decrease the conversion of acetyl-CoA to malonyl-CoA. That will decrease malonyl-CoA levels. On the other hand, whatever malonyl-CoA that are already there, they will be decarboxylated to acetyl-CoA, done by malonyl-CoA decarboxylase. That is also activated by AMPPK, because AMPPK is going to keep malonyl-CoA decarboxylase active, thereby further decreasing malonyl-CoA levels. So that means AMPPK is going to decrease the formation of malonyl-CoA by keeping acetyl-CoA carboxylase inactive, and also it is going to increase the degradation of malonyl-CoA into acetyl-CoA, thereby by keeping malonyl-CoA decarboxylase active. So overall, AMP-PK, it is going to totally decrease malonyl-CoA levels. When the malonyl-CoA levels are totally decreased, which is shown in number 2 reaction here, so its negative effect on CPT1 is not there. So the CPT1 here, CPT1, carnitine palmityl transferase 1, negative effect is not there. So fatty acids are easily flowing into the matrix of mitochondria, and they will undergo oxidation to give energy needs of the skeletal muscle and cardiac tissue. So like this, the regulation of beta oxidation of fatty acids, it is all centered around malonyl-CoA concentration and its effect on CPT1. Anything that increases the concentration of malonyl-CoA will decrease beta oxidation, which is done by insulin under fed condition. And anything that decreases the malonyl-CoA concentration will increase beta oxidation, because CPT1 will be free to work, and that is done by epinephrine, glucagon, and also during exercise AMP, which is increasing AMP protein kinase, AMP activated protein kinase. Okay, so two enzymes that you need to remember here, one is acetyl-CoA carboxylase, other is malonyl-CoA decarboxylase. When acetyl-CoA carboxylase is inhibited, malonyl-CoA levels will decrease. When malonyl-CoA decarboxylase levels are when malonyl-CoA decarboxylase is activated, so again malonyl-CoA levels will decrease, because malonyl-CoA is diverted into acetyl-CoA formation. So overall, fatty acid oxidation will be regulated in this way. That's all about regulation of fatty acids. Thank you.